Hi, everyone. First of all, thank you so much, Fernanda, for putting this together. I know it has been a lot of work. Can you hear me? Yeah. Thanks, Fernanda, for putting this together. I know how hard you have worked with your team, so thank you so much. And I hope it creates, as Fernanda is working so hard for, bonds between Massachusetts and Chile. It is my great pleasure to introduce Manolis Kellis, who is my amazing boss. I had to write it because he has so many talents that I couldn't forget any. Manolis is a very unique and extraordinary person, a brilliant scientist with extraordinary vision. And a testament of this is that he has been invited to more than 50 talks, has more than 200 scientific publications, and has received many awards. Because of the time, I will only mention a few. He has the Presidential Early Career Award in Science and Engineering by the US President Barack Obama. He was awarded as the top 30 principal investigators in the Future Genome Technology magazine, top 35 innovators under the age of 35 Technology Review magazine, National Science Foundation Career Award, one of the 3 young scientists honored as the next generation in Biotech, Museum of Science, and many others. I remember when I met Manolis, I was so impressed with the speed that he does everything, the speed of thinking, of action, that I asked him, Manolis, are you sure you are human and not a machine? And he just laughed and never answered that to me, so I'm still not sure. I'm still testing that hypothesis. In addition of being brilliant and always optimistic and full of energy, as you probably will see in his talk, Manolis is also an extremely kind and caring person with everyone around him, mostly his family, and all of us, his group, which he constantly says that we are his scientific family. He frequently opens his home to us, organizes field trips that are amazing, and it's very impressive to see a faculty that is at his level to always care for the people in the group, if they are all well or not. And when anybody is sick or he notices they need help, he immediately is there. He's originally from Greece and is fully trained at MIT. This is the MIT day, and you will never find a more MIT person than Manolis. He did his undergrad here, his graduate studies here, and is a faculty here. I think the only thing he didn't do is the daycare, but maybe there wasn't any at that time. Yeah, you know. Yeah. So I don't know if being Greek or probably that's a reason, but he has so many interests and knows everything. So it's really fun and challenging to talk to Manolis. He can talk about aging brain, cancer, any other complex subject, evolution, and to the meaning of any word. And this is what keeps me working with him, because it's always a source of knowledge and challenge at the same time. He has been working in the field of regulatory genomics and epigenomics for the last 14 years. He pioneered the first methods for the novel discovery of regulatory motifs using genome-wide comparative genomics and applied them systematically in the human, fly, and yeast genomes to discover dictionaries of known and novel regulatory motifs across the genome. I should let him talk now, because he can explain way better what he does than I can. Manolis, it is such a great honor to have you today here at ChileMass, which is Chile my lab, of course, and science my other lab. So both together is extremely amazing to me. Please take it away.
Querida Patty, no tengo palabras para decirte cuánta suerte tengo de poder trabajar contigo y de poder hablar con toda la gente que está aquí. Yo soy de Grecia, pero tengo una esposa de Francia y estudió el español durante 3 semestres aquí en MIT hace 23 años, pero es una lengua muy linda y muy acerca de mi corazón. I'm going to switch to English, just for the broader audience. So it's truly an honor, a pleasure, and I'm humbled by your very kind invitation. I'm humbled by the very kind words that Patty said. The last time that Patty was on this stage, it was with President Rafael Reif, receiving the most prestigious award that MIT has for a member of our community. This is the Collier Medal, named after the policeman who was very horribly killed during the terrorist attacks of Marathon Day here at MIT and in Boston. And the award represents really a member who brings the community together, who stands for community, who serves as a pillar for our community. And I cannot imagine a better recipient for this award. And what they told me from the award committee is that they had never seen a stronger nomination, and it was not even a question. So Patty, a huge thank you to you, to your heart, and I think the Chilean blood running in you speaks a lot for that, so thank you. So on my day job, I'm a professor in the computer science department, believe it or not. And yet what I will tell you about today is human disease and human medicine and human disease circuitry. How is that possible? I would say that there's no other way. The only way to truly understand the human genome, to truly understand personalized medicine, and to truly pave the path for the future of therapeutics, for the future of a healthier society for all, for the whole world, the only way to do that is data integration. It's large-scale data generation, it's systematic manipulation, and it's a systems thinking, which is uniquely representing what MIT stands for. MIT knows no boundaries of departments. MIT is a place where everyone can thrive from the whole world, and everyone can truly make a difference, no matter what label you have, whether your label is undergraduate, whether your label is biology or computer science or professor or dean, it does not matter. There are no boundaries, and that has truly served our community so well. So I'm extremely privileged to be a member of this community and to be at the head of this unbelievably kind, generous group, several members of which are actually here at the audience. So Yosuke and Ben and Shushen, and I don't see because of the light, but these are the folks that I glanced at on the way here. So anyway, thank you guys for being here, and thank you for being just such a kind group. So What's really unique about the current phase that medicine is going through is that for the first time in human history, we have causality. Epidemiology has always been about correlation. If you drink coffee at Starbucks, you are more likely to not have cancer. Now, is this because the people who can afford a coffee at Starbucks also have better healthcare system? Or is it because really the coffee at Starbucks is so much better? So correlation does not imply causation. And there are so many things in healthcare which are completely intertwined with socioeconomic status, with ethnic background, with what society you live in, with your zip code, and so on and so forth. And that has been true throughout the history of humanity. Even before we had zip codes, we always have looked at correlations. But for the first time, we can look at causation. Why is that? Because of genetics and because of genomics. So what I'm going to tell you about today is how we can leverage the genomic revolution that has happened over the last 20 years to transform the face of medicine for generations to come. So how do we dissect and how do we manipulate disease circuitry at single-cell resolution? Genetics is not a recent invention. Humans have understood genetics for thousands of years. We have bred animals for their fur. We have bred corn for its food. We have bred animals for their behavioral traits. And we have understood that inheritance of eye color and face shape and height have always been true in humans. But it's not until Gregor Mendel carried out experiments in a model organism, the peas that he was using in his garden, that we truly understood that inheritance is discrete, that there are units of inheritance, which we now call genes, and that these units are assembled and inherited independently between distant locations of your genome. And these rules of inheritance would be to transform the face of human medicine. But they remained ununderstood for 50 years. There were only 3 citations to Gregor Mendel's work for 50 years, and an unopened copy of his book was sitting on Darwin's shelf after Darwin was dead. Why? Because humans observe that variation is continuous in human populations. That in fact, eyes are not simply of 3 colors, they're a continuum of colors. Hair, similarly, skin, similarly, height, similarly. Every aspect of human physiology, of human behavior, of human psychology is in fact a continuum. We like to give them discrete boundaries to call someone Alzheimer's or schizophrenia or diabetic or obese, but those boundaries are artificial and human physiology is continuous. So how do you reconcile these views? On one hand, I'm telling you that genetics is discrete. On the other hand, I'm telling you that humans are continuous. The way we reconcile this is Ron Fisher's work in the early turn of the century, in 1900s, basically showing that even 5 Mendelian loci, even 5 genetic locations that are inherited with 1 0, can still give the semblance of a continuous distribution of height. Why? Because of polygenicity. And the Mendelian traits that Gregor Mendel had discovered are still there in the human populations. There are many strong effect genetic regions that will have a big impact on inheritance. And those were discovered very, very rapidly through, in fact, subtle deviations from Mendel's laws of independent assortment. In fact, the closer 2 genes are on a chromosome, the more likely they will be to not segregate independently in the population. And complex traits, however, took many more decades. Why? Because the genetic variants that impact them are extremely subtle. They have very small effects. And if you ask, how often do we find variants with small effects versus large effects in the human population? The answer is overwhelmingly weak effects. Genome-wide association studies have found hundreds of thousands of genetic differences, each of which is robustly, causally associated with disease. But very, very few of them do we understand how they work. So in this talk, I will describe how my lab and how many others in the community are working together to try to understand how these loci work and how we can translate these genetic and genomic findings into therapeutic insights. On one hand, the promise of genetics is that you can look at a plot like this, which is known as a Manhattan plot because of these tall skyscrapers, and interpret that, in fact, there's something going on here that is responsible for disease. There should be a little, perfect. So in this region, every dot is a genetic difference between each of us. And the height of the dot is how significantly associated it is with body mass index, a measure of obesity. That basically tells you that there's something going on in this FTO locus that was previously named fused toes O, and then was promptly renamed fat and obesity associated, because that locus clearly has a causal role on obesity. If you inherit those genetic alleles associated with obesity, you're going to be much heavier than the typical person. So that gives us a handle for perhaps understanding disease mechanism, finding new target genes, finding new therapeutics, and enabling precision medicine and personalized medicine. The challenge, however, is in 93% of cases, those genetic differences do not impact disease. FTO, giant locus, everybody was expecting to impact the gene. Not at all. There's not a single protein-altering variant here. There are 89 common variants that are co-inherited, none of which is in fact perturbing a protein. So that basically means that we don't know the target gene, we don't know the causal variant, we don't know the cell type of action, we don't know the relevant pathways, and we don't know the mechanism. So that's what we're about to set out to find. What we showed in my own lab is that the true targets of these locus are not the FTO gene at all. They're instead two genes that are sitting 1.2 million nucleotides away, 1.2 million letters away. Several genes away are the true targets. And without understanding this, we would never be able to create therapeutics against obesity, diabetes, Alzheimer's, schizophrenia, and so on and so forth. So here's the path that we're taking in our lab. Number one, we're starting with genetics of common variants and rare variants. Number two, we're then profiling. So this is the starting point. So the first step we're doing is profiling how gene expression changes, how the 20,000 genes in our cells are in fact changing in every cell type in response to both the genetic variation and the disease variation. And we're also studying how the epigenome is changing. So what is the epigenome? The epigenome is the packaging of the genome. So the DNA is packaged around these nucleosomes. And you can modify these packages. Kind of like when you're packing your house, you put a label on the box saying living room and saying, you know, dining room. So that when it's time to sort of open up in the dining room, you open only the boxes that say dining room. The exact same thing happens in the genome. Every tag has specific regulators that only turn on in liver. And all of the boxes carrying the liver tag only get opened for liver. And the neuronal tags get opened for neurons. So we profile how these tags change now, not just across tissues, but also across individuals. Every one of us has a unique genome. Every one of us has a unique environment. Every one of us has a unique set of behaviors and traits and nutrition and pollution exposure and epigenetic inheritance and socioeconomic status and ethnic background. And all of these are in fact influencing both your gene expression levels and your epigenome. And the epigenome uniquely tells us about the circuitry of every one of your cells. Raise your hands if you're with me so far. Awesome. Who feels that they've learned something? Awesome. Who has a right hand? All right. All of the people with right hands are following, which is great. If you only have left hands, you can raise that one too. All right. So we're profiling all that. Why? Because we are primarily a data integration lab, and you can't integrate data without data. So basically, we then integrate all these data by developing methods like deep learning, like causal inference, like Bayesian networks, like contextualized learning, and then we use all that to understand what are the driver genes the driver control regions, and the driver cell types that are underlying the disease. And then we validate those predictions by perturbations. There's two ways to get at causality. The first is genetics. The second is intervention. So we basically are able to do interventions, number one, in mouse, and number two, in human cells. So we basically can use model organisms to understand the multi-tissue and multi-organ effects, and then to understand the cellular consequences, we can basically study human cell lines, and to understand the organ-level interactions, sometimes we can actually use human organoids, also known as organs on a chip, which basically allow you to study the vasculature, the immune system, the nutrient response, etc., in the context of a multicellular culture. So our first step is basically large-scale data gathering. So how do we do that? We have this experimental group that is absolutely outstanding. So Kiki Galani and Lee Lun Ho and Ashley Renfro are just amazing individuals. And they have basically profiled at single-cell resolution thousands of human samples across tens of millions of human cells. Across Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's disease, Alzheimer's with psychosis, schizophrenia, bipolar disorder, Down syndrome, autism, post-traumatic stress disorder, addiction, aging, cardiac disease, obesity, and exercise. And in all of those different conditions, we're asking how are the genetics, how's the environment, and how are the cell-cell interactions affecting every cell type, every RNA, and every gene regulatory region across multiple tissues and multiple regions of the brain. The second step is understand the circuits through which these genetic variants are acting. How are they affecting transcription, gene expression? How are they affecting methylation in the epigenome? How are they ultimately impacting disease, and how is all that affected by the environment? And we're building deep learning models that can understand the language of the genome and the language of the epigenome by basically understanding how the genome itself, how the cell itself interprets its own DNA, understanding that programming language, understanding the words, the sentences in hierarchical constructs, and then using all that to now start prioritizing individual genetic variants. So going back to that first slide that I showed you of the FTO locus, we can now ask, how is that region impacting obesity? Because simply telling you, sorry, you have a predisposition, how good is that? What we want is therapeutics. What we want is to understand these pathways so that we can then go and manipulate these pathways. Raise your hands if you're with me so far. Yes? Awesome. Good. More people grew hands in the meantime. This is great. So how is this FTO locus acting? Notice the FTO gene doesn't even feature here. We have narrowed down these 50,000 letters, 50,000 nucleotides into a single letter, a T to C mutations. A single mutation from T to C in this AT-rich motif makes that this protein can no longer bind. What does that protein do? We showed that that protein is a repressor of those two genes, sitting 1.2 million nucleotides away and 600,000 nucleotides away. These then impact thermogenesis. As a Greek, thermo is heat and genesis is generation. So generation of heat. What is thermogenesis good for? Turns out that your fat cells are your best friend. They're not your enemy. They store all of the excess calories in your diet so that you have them for a rainy day. They're also preventing all of your organs from suffering from the lipids circulating otherwise in your body. So they are also able to burn your excess calories by depolarization of the mitochondrial membrane and loss of energy as heat. Your cells are able to do that. My cells are not able to do that. They are stuck in the off position. My cells, every calorie that I don't burn by the end of the day will be stored as fat because I inherited two risk alleles from both my mom and my dad, thank you both, for obesity. So that basically means that I am starving right now, and I was starving all day yesterday until 6 p.m., and I went running this morning, and I went swimming yesterday in order to not be obese. So basically, I have to sort of make up for my genetic makeup. And the reason is that my cells are basically stuck in the lipogenesis position. So what can we do, number one, about me, And number 2, about everyone in here who carries those alleles. But number 3, much more importantly, about every person in the planet who suffers from obesity, whether it's genetic or not. And I'm not here to edit genomes. I'm here to basically understand how the circuits are working so that we can then modulate these circuits and reverse human disease. Understanding these circuits means we can manipulate these circuits. And that means that we can increase or decrease the upstream regulator. We can increase or decrease the downstream target genes. And to validate causality, we can even use CRISPR-Cas9 genome editing in primary cells from the adipocyte of obesity risk individuals. I will not disclose whether these were my cells. They were not. And show that, in fact, a single letter can, in fact, alter thermogenesis. Here, we're able to completely restore the process of thermogenesis. And we're able to go into mice and put a dominant negative form to repress these genes that are overexpressed in obesity. And guess what? These mice lose 50% of their body weight. And when you put them on a high-fat diet where normal mice gain weight, these mice burn all the calories. They're just burning calories in their sleep. When they're awake, when they're walking, when they're sitting, when they're resting, when they're sleeping, they're just burning calories through thermogenesis. When you open up these mice, their fat stores are completely depleted. There's not a single ounce of white fat. It's all brown fat. It's all red, healthy tissue. So how can we do that for every single human trait? The first thing we need to do is understand the epigenome systematically. And we have built resources, we have built these references that the whole world is using to understand the epigenome. And that allows us to understand the circuitry, the pathways, and the tissues of action of disease. Number 2, we want to profile thousands of individuals at single-cell resolution to understand how these cells are changing across Alzheimer's and all of these other conditions. Number 3, we want to understand the circuitry of those cells during disease progression. In the case of Alzheimer's, we've basically sorted all of these cells based on how AD-like or non-AD-like for Alzheimer's is their transcriptome. And then we're finding the genes that are causing Alzheimer's that are acting early, middle, or late in the progression of disease, enabling us to build therapeutics that can now start acting earlier in the disease. We need to understand the 3D placement of every cell in the context of tissues to understand how they're interacting with amyloid pathology, with neurofibrillary tangles, with neuroinflammation, with all of these different pathways that are then impacting those cells in their spatial context. We need to develop tools for systematically modulating these in every tissue and every cell type in the body to understand that causality. So we've developed such modular and programmable constructs that we can now differentiate into every single cell type and understand how we can turn on or turn off regulatory control regions, how we can turn off genes, and how we can change individual letters of the DNA. And we have also developed massively highly parallel assays so that in a single experiment we can carry out 7 million measurements. This is a computational student in my lab who basically developed this new technology that allows you to just chop up regions from the genome, insert them into self-transcribing, self-reporter assays, and then test every one of these constructs across the entire genome in a single experiment, and then narrow down the letters that are responsible for gene regulation so that we can understand genetic variants, so we can find the upstream control regulators, and so that we can understand their conservation level. We also need to understand how patients vary within the clinical record. We have basically built these multimodal approaches for understanding the state of a patient, for personalized medicine, for understanding subclasses of individuals based on multimodal inference across doctor notes, across prescriptions, lab tests, etc. So we can complete these records and start associating them with gene expression differences. Yosuke, who's in the back of the audience, developed a new way of understanding the building blocks of disease, the hallmarks of disease, taking complex traits and then partitioning them into a palette of changes that every person has. We shouldn't be thinking of Alzheimer's as a single monolithic disorder. No, Alzheimer's is a combination of multiple individual pathways. And everyone who carries Alzheimer's has a different manifestation. So what Yosuke showed is that we can, in fact, discover not only unique hallmarks of disease, but also unique combinations in every patient, grouping patients into different subtypes for which we will recommend very different treatments. In the front page of MIT, at mit.edu, you will see today our study on the effects of exercise and obesity across multiple tissues. So we basically studied in mice and also in humans how obesity, nutrition, and exercise are in fact pushing against each other across dozens of pathways and across multiple tissues. We cannot study cells in isolation. We have to understand the interplay because many of these disorders are systemic. They're affecting your entire body. And Ben Lengerich, who's also here in the audience, basically recently published a paper showing how if you go in the clinical record, what you see is not a monotonic relationship between biomarkers of disease and disease risk. Instead, you see these very sharp thresholds. And the reason for that is that these biomarkers themselves are impacting the decision for intervention, which then changes the risk and changes the risk again. So as we're building AI models, he basically developed a method that allows those AI models to be more intelligent and to not simply say, oh, great, all we need to do is increase the creatinine level for that person and they'll be doing great. No, what we need to understand is what are the components that are contributing to this so that we can then go into clinical records and carry out massive artificial intelligence data mining to understand these pathways. The next direction that we're taking right now is understanding therapeutic development itself, understanding how we can build drugs, how we can design drugs. And we're using deep learning to design three-dimensional chemicals that will bind in the pockets of all of the domains that are associated with these hallmarks of disease so that we can then understand how to intervene in a precision medicine, not just, oh, here's a drug that we don't really know how it works, but here's a molecule specifically for this pocket of this regulator that interacts with this protein. So I see the future of medicine as transformative using all these technologies. On one hand, today, we have more diseases than we know how to name. In my own family, we have a predisposition for obesity, for cancer, for stroke, for diabetes, and obviously for hair loss. My own predispositions are for obesity, blindness, and cancer using my own genome. Every one of us in this room carries mutations that can tell you about your risk of disease and can tell you what interventions you should be taking now in order to ameliorate your future symptoms. So how do we leverage all that? There's also a changing environment with pollution, differences in nutrition, with fast food, with a sedentary lifestyle. There's systemic disorders. As I mentioned, obesity is affecting every tissue in your body. It's making every disease predisposition worse just by having that obesity component. And there's also increasing pathogens, infections, and all of these infections are leading to immune dysregulation in your own body. And of course, many of them are in fact integrating to your genome and eventually leading to cancer. And the increasing lifespan basically means that the prevalence of Alzheimer's is projected to double in the next few years. And many new diseases will start appearing that we haven't seen before. So how do we address this major challenge? What we need to do is transform pharma. Instead of being always surprised when the symptoms first appear, we want prognosis, both for Mendelian genetics and also for polygenic scores. Instead of constantly making the wrong judgment for infants, for adults, for elderly, and then giving the wrong treatment, we want better biomarkers. We want multimodal diagnosis. Instead of only treating the manifestations of the disease, what we want to do is address the root causes of the disease, understand these causal hallmarks of disease. Instead of thinking of diseases as monolithic, like Alzheimer's, type 2 diabetes, we should be understanding the heterogeneity of both the symptoms and the causes across individuals. To understand the components of disease, for Alzheimer's, it's amyloid beta, and tau, and inflammation, and lipid, and cholesterol dysregulation. We need to stop having silos, both in medical departments and in pharmaceutical departments. Instead of just having every tissue be its own building, what we need is to understand the interplay between these tissues, the commonalities in the pathways, the sharing of molecular mechanisms across different diseases and across different tissues. Every disease affects multiple tissues and multiple pathways, and every tissue is implicated in multiple diseases and multiple pathways. Instead of treating too late, we need preventive, personalized interventions. So how do we go about this? How do we personalize medicine? What we want to do is, number one, embrace polygenicity. Every single trait has thousands of variants that are associated with it. Number two, understand the convergence of these thousands of variants into a small number of common pathways, these hallmarks that I'm mentioning. Understand how to manipulate each of these separately. Transform clinical trials. Instead of the endpoint being these monolithic disorders, have the endpoint of every treatment be the impact on that pathway with specific biomarkers. Understand how we can individualize treatment by combining the specific pathways that are dysregulated in every person. For every patient, understand the unique combinations of burden that they have across each of these pathways at the genetic level, the epigenomic level, the transcriptomic level, the proteomic level, lipidomics, and so on and so forth. So I want to make a call to action to all of Chile and all of Mass and all of the world to basically come together across different disciplines. What we need is a multidisciplinary approach. What we need is computer science with machine learning and deep neural networks to understand the DNA code, the circuitry, to integrate all the big data. We need biology for high-throughput profiling and high-throughput manipulation of these biological systems. We need chemistry with new libraries, new approaches for high-throughput synthesis, for modular synthesis. The Nobel Prize yesterday was specifically this. It was modular synthesis of chemical compounds. We need biotechnology for rewiring, for delivery. We need finance for long-term, 10-year, 20-year horizons with biobonds that are issued, that mature, because the current financial incentives for venture capital in pharma is completely off. Everybody gets out before phase 2 clinical trials are back. And the reason for this is that we're thinking in a very short time horizon. We need new partnerships across pharma. We need empowerment of patients. We need combination of cohorts across hospitals. So I'm going to stop there and basically say how privileged I am to be in this position, to be at MIT. My group has had this amazing success in sort of populating some of the best departments in the world with top trainees. We've had hundreds of thousands of citations, dozens of active grants. I'm so grateful to the funders, to the NIH, to this incredible community that we're part of. And we are just so lucky to be here. So basically the message is join our efforts at MIT across computer science, across computational biology, big data, machine learning, experimental validation. We're having a lot of impact, having a lot of fun. There's lots of other faculty members. I'm only one representing a very large community. And this is also just a unique environment for innovation across academia, across hospitals, across industry. You basically see all of these biotechs that are founded within a square, like half a square mile from here, a square kilometer from here. There are so many different innovations that are leaving academia and entering the clinic. So I hope that you are all so excited to be part of this incredible community. I am so excited to sort of see so many of you here coming together, and I really hope that across disciplines, we can truly make a difference. So I'll stop there and thank again Patty for the wonderful invitation, the organizer for putting together such a great meeting, and all of you for your attention. Thank you so much.